Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about heredity, and in particular what I like to call Mendelian genetics, sort of giving honor to the great biologist Gregor Mendel, the Austrian monk, who gave us some of the most fundamental patterns of heredity that are still with us today. And I wanted to start off our conversation today with a look at, let's go here, a look at this particular picture. And some of you may know that these are my daughters. And one of the things about the reason I put this picture up is, one thing, I'm proud of them and love them. But the thing is this picture was taken sort of circa like 5th grade. And I have to believe that every parent at some time in their life looks at their children and they ask this fundamental question. I think it's within all of us. It's like, where did that trait come from? How come this daughter has curly hair and this one has straight hair, and they seem to have the same nose, they seem to have the same smile, different chins, different eyebrows. You could sort of break it apart trait by trait and look at this. Now, the thing is, you might think, well, this, you could trace it back. This came from this parent, this came from this parent, but how does it work? Because sometimes both children look like one parent, or they don't look like any of the parents, or they look like the grandparents. And so one of the things about heredity is, for a long time, people didn't really understand how it worked. You can imagine it's a difficult thing to sort of untease. And so, it's often been said that there's as many people proposing ideas about heredity as there were different ideas about how it happened. And so, not to say that we have a complete understanding of heredity, but we have some basic ideas. And those basic ideas came to us not recently. These basic ideas came to us in the 1800s. And so this particular video, I want to take us back and look historically, as I often like to, at some of the great and wonderful biologists that sort of uncovered some of these wonderful and interesting concepts. And the reason that I do that is not only because they're contextually important for you to understand, but I like to give honor to those individuals, and I'd like to sort of get into the mind of those individuals. I'd like for you to get into the mind of those individuals. So the intention is, I want you to someday emulate these individuals, and maybe someday you'll be somebody will be making a podcast about you. And so, to that end, you look at different traits. You can look at eye color. People love to talk about eyes, and you're like, I wonder how these traits get transmitted. And so, obviously, they're from our parents. And what's actually being passed, as you know, is our DNA from one organism to the next. But what's interesting is people have been thinking about this for a long time. Like most people thought, if they were giving it any thought at all, and it makes perfectly good sense, that if a blue bird were to reproduce with a yellow bird, sort of like paint, if you mix blue paint with yellow paint, you're going to get green paint. And so this is what we'd call the sort of the blending hypothesis about how heredity works, and that this is why you would get different colors. But the truth is, when you looked at this blending hypothesis more closely, it proved to not be as accurate. And so there's all kinds of examples that will come up in this discussion and in future ones that blending isn't exactly the way it works. I mean, you could have black mice producing only black mice. You can have black mice producing black mice and white mice, and then you could have instances of gray, and you would think, well, once you have gray, that's it. If gray and gray reproduce, they're only going to have gray mice children. But the truth is, the gray mice in the first generation, when they have children in the second generation, they can produce black mice and white mice and gray mice. So you're like, wow, it seems kind of random. It's like, it's hard to know, hard to predict. It is difficult to predict, but we're going to make some attempts. And in one of the great brave individuals that I want to highlight in this video on inheritances in the garden is, I think, one of the great biologists, not because of just of his contributions to biology, but because of his bravery, because of his creativity, because of his dedication and his commitment to for discovery. And so I want to give it up for the Austrian monk Gregor Mendel. And Gregor Mendel um, was a monk, and sometimes he's referred to as the father of genetics, literally and figuratively. Um, what he did was he was working with pea plants, as you may be aware of, and what he did is he grew them for many years. And what he did was he took great notes. He kept great documentation of the patterns that he would see in each generation when he planted the peas. He would take copious notes about patterns that he was seeing. And after a while, these patterns began to emerge, and he felt confident that there must be some reason, some logical reason why inheritance works the way it does. And today we know a lot about inheritance, a lot more than Gregor Mendel did in the 1800s, but it's because we stand on the shoulders of people like Gregor Mendel that we're able to see as far as we do. So I give it up to Gregor Mendel, one of the greats of all time in biology. Although his story began rather humbly. He grew up on a small farm in what is now the Czech Republic. We believe that this was his home. And growing up on the farm kind of gave him a sort of leg up on being able to work with plants, and he was familiar with nature, that sort of thing. And so later in his life, not to go over every detail, though it's interesting, is that he entered the monastery, 1843, and we believe this is the monastery right here. And as it turns out, he studied at the University of Vienna. And one of the things I want to emphasize is when he was in college was the fact that he was learning by a physicist who encouraged an experimental approach to solving problems, sort of the scientific method. In other words, you have observations, you have a hypothesis, you sort of follow it up. It's deductive reasoning, and you sort of test and see what's happening, come up with conclusions. And then he was also influenced in a way that he wanted to apply mathematics to science. And so this was an emerging field of mathematics, kind of new at this time, statistics. So the interesting thing is he was a little bit more knowledgeable than the next person in the 1800s about mathematics and statistics. And obviously he knew a lot about gardening, and he studied botany as well. And so as it turns out, this idea of statistics and gardening come together, and they sort of gelled into what we now know as Mendel's experiments on heredity. And so you may be familiar with the fact that he was working with pea plants. And so what's interesting is after he graduated, he was a teacher, and he was a monk, I mentioned. And interestingly enough is monks in general, not just Gregor Mendel, have a long tradition of breeding plants. And in this case, the monastery where he was stationed was breeding peas. And so he fit right in. He's there in the garden, on the ground with his apron, and he was crossing pea plants together. And so one of the things about pea plants is that when he started breeding them in 1857, a while ago, he thought that he might be able to see a pattern in what was happening. And in terms of when I say pattern, what was happening is you can look at different traits of a pea plant. Because we're not so familiar with pea plants, sometimes we may not even think of any traits to consider. Like for humans, you can think of all different kinds of traits. Dimples or shapes of earlobes or hair color or eye color. You can just keep going with this. But pea plants have traits too. And they have interesting sort of contrasting traits. Like in other words, like their stems, some pea plants are really tall and some are kind of rather short, so tall and dwarf. Some plants have these pretty purple petals. Some have white. Normally I think most of us think of a pea seed as being green, but some pea plants have yellow. And so they have these interesting, very easy to observe traits. And so this is where he's coming from. He's sort of asking simple questions. What would happen if you crossed a pea plant with purple petals with one with white? What would the next generation look like? How does heredity work? And what are the possibilities? Maybe all of them are purple, maybe all of them are white, maybe there's some sort of blending, maybe they're sort of light purple in color, maybe it's 50% purple, 50% white, maybe it's random, maybe that, no one knew. And so one of the first things I want to talk about before we get into those crosses is that, in order for a pea plant to cross, sexually, you have to know a little bit about floral structure. And so what we're looking at here is a typical flower. These structures right over here are the stamen. And the stamen are composed of an anther held up by these filaments, and there's many. That yellow powder is pollen. That's the male gamete sperm. This structure right here is the pistil. It has a sticky stigma, a style, and the ovary down below here. And so a plant can either self-fertilize, in other words, pollen and egg from the same plant, or it can cross-pollinate, meaning pollen from one plant to another. So Mendel was obviously aware of this, and he was able to cross plants and also self-fertilize them. Here's a little bit of a diagram of it, what I was talking about. I wanted to start off with the real flower, but a diagram is helpful too. Petals are the sort of modified leaves that are colorful, that are attractive to pollinators. Here are the stamen, right here. These are the anthers in yellow. That's where the pollen is produced through meiosis. And the pistil is this female structure in the center here, composed of a stigma, style, and enlarged ovary. The sepals, as you may know, are these base leaves that actually enclose the petals, and at night they kind of close up. If you're maybe familiar with a rose, these sepals down below here, they protect the flower. And so another diagram, different perspectives help you to understand. So here are the stamen, the male structure, anther and filament. Filaments hold it up so that the wind can help pollinate better. The filaments can also be kind of different from flower to flower depending on the pollinator. The female is called the pistil, which is composed of this sticky area called the stigma. Why does it have to be sticky? Because the pollen that's produced, there's a lot of it, and if it can stick to the stigma, it has a better chance. And what happens is the pollen then sort of like an ant bores a tunnel down the style around the ovary, and it goes into this little hole called the micropyle, and it enters into a structure called the ovule. And inside the ovule, sorry for the terms, inside the ovule is a reproductive structure called the egg, and it's the female gamete. So there it is. So meiosis in the female structure, the ovary is producing the eggs. It's actually the female gametophyte, as it's referred to, is actually something called the embryo sac, but we'll discuss that in a different video. So this is what we'd call self-pollination, where the same flower is producing the pollen and it's fertilizing the egg down below here. Now this will eventually become, you ready for this? The seed. So this is where the seed is forming inside the ovary. And then the ovary will enlarge like this, and there could be more than one seed. And so the enlarged ovary is the fruit that we like to sometimes call the pea pod, and then inside the pea pod are the seeds, which is the next generation. So these are the children. When you plant the seed, it'll eventually grow up into another pea plant. Another diagram. You might notice, just to emphasize this, I was calling the female structure right here in the center, I was calling that the pistil. It's also known as, sometimes there's more than one term, carpel, but it's still stigma-style ovary. Here's the ovule, and then inside right in here is the female reproductive gamete, the egg. Here are the sepals, here are the stamen over here producing pollen. Very nice. So sometimes a diagram that doesn't have any labels on it is informative, and so I can ask you some questions about this. I can say, what number of the structure is where the pollen sticks? And you'd say pollen sticks right up here, number 4. So that the answer would be 4, sticky stigma. I could say where does male meiosis occur? Like the formation of pollen that occurs in 3. What number is the structure that attracts bees to the flower? And the answer to that would be 1, the petals. Where does female meiosis occur? That would be 7, inside the inside the ovule, right in here, producing the egg. What structure will eventually become the fruit? That would be 6, the ovary. Let's see, what number makes up the whole female structure? And that would be 8, and that would either be called the carpel or the pistil. Another advantage of working with peas, other than the fact that they're easy to work with in terms of like recognizable characteristics and sort of traits that can be contrasted, like tall, short, or purple, white, is that they make a lot of offspring. They make a lot of peas, which is great for statistical analysis. But you can also have really easy control over them. You can determine who's going to mate with who, in terms of like you could cut the anthers off of a plant and then make sure that it's only crossing with another plant, that sort of thing. And so what Mendel basically did was, if you wanted to cross, and this is the way we sort of set this up, if you wanted to cross like purple with white, and so that's the trait that we're going to consider, what you would do is, it's not shown here, but I'm going to like draw it. It's hard to see it right here, but the anthers right there are producing the pollen. So what you'd want to do is get some pollen, and you'd want to transfer it. You're like, what are you doing? Like with your finger? Now you would probably use like a paintbrush to do that, and then you would literally rub that on the stigma of the other flower, and then the pollen would just naturally work its way down. And what would happen is, when you fertilize, like if we were transferring the pollen from this purple one to the white one would literally produce a big pea pod like this, and then there's be seeds inside. So these are the children. You can go the other way too. These are the children of this cross. So these are the parents, these are the offspring. And so the way we talk about that is we say that this first generation is the pea generation, not pea as in pea plant, but parental. This is the parental generation. These seeds inside here is the first generation, and we call them the F1s, which is subscript 1. So it's philia. Write that out, philia. And I hope I spelt that right. Philia. I hope. I apologize if I didn't. But philia is kind of French for daughter, or for fille. So it's his first generation, or F1s. And so you're like, okay, what was the question? What happens if a purple crosses with a white? What will it look like? Will they all be purple? Will they be sort of light purple? Will they be white? Blah, 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 blah. There's no way to know it until you do it. And then you open the pod, and then you take the seeds, and you literally like plant them in the soil. So there it is. And then when it grows up like this, produces the leaves like this, and then, here comes the flower. And if the flower is purple, then you can say, well, that's interesting. Purple and white, when you cross them together, it makes a purple flower. It seems like the white goes away. And you're like, well, is that just that one seed? And you can plant them all and see what happens. So this is what he's doing. And you might be like, well, why is he doing this? Because he's trying to look for patterns of heredity to sort of see what's governing this. And so here's a picture of real pea plants. Some of these still have the petals on them. Usually the petals fall off once the ovary becomes enlarged. And so this is your typical pea pod. These are the seeds inside, or the F1s right there. And so this is what I was trying to describe to before about paintbrush. So this would be like the hand of Mendel right here. So what he could do is, take some pollen from one plant and put it over here to the other. And this way you're transferring pollen from one plant to the other, and then the pod is going to form. And then you take the seeds and then you grow them, and then that's the first generation, or the F1. And then if you continue this, if you let these plants reproduce, then you would have an F2, and you have an F3, and you keep going with that. And so typically what Mendel did was he cross-pollinated, or what are called true breeding varieties. So he had plants, for example, that were always tall. And when you cross tall ones with tall ones, they made tall children, always tall, 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 for generation after generation. This plant looks a little shorter. Every time you self-pollinate that and a pod forms, it produces short. So it's like pure short, pure tall. He did that for a couple of generations. And then he set the cross. And he's like, okay, now I'm going to cross tall with short. So I'm going to do tall with short and see what happens. And you're like, what do you mean see what happens? Well, if you take the pollen from the short one, you put it over here to the tall one, eventually this is going to form a pod. And then here are the seeds. These are the F1s. This is the parental cross. This is the F1. And then when you plant these, what's it going to look like? Well, I don't know. It remains to be seen. It's a mysterious. They could be all tall. They could be all short. They could be medium. One never knows. But the point of this is the children, he called them hybrids, because you're hybridizing. In other words, like you're mixing a tall and a short. And these are the hybrid offsprings. We're familiar with the word hybrid today because we have these hybrid cars which run on battery, some gasoline. So there's sort of a mixture of those two energy sources. So the term hybrid means like a cross between two different things. So tall and short, these are the hybrid seeds. And these are the P generation for parents, and the F1 are down here. So what does he do? He's hybridizing these, and so he's determining some patterns that are emerging. And so check this out. When he crosses in the parental generation pea plants that are purple with white, every time he does this, and let me just sort of act it out if you don't mind, every time he crosses the pollen and a pea pod is formed, every time he plants the seeds in the F1, the hybrids, they're all purple. Every time he does this, purple and white, purple, purple and white, purple. The children are always purple. So don't jump to conclusions and say, well, purple is a darker color, and so therefore it's probably covering up the white. No, purple. Not even light color, just as pure as the parent. So what's interesting about this is that if the blending, remember we talked about originally, we said that blending, like if you mix the yellow bird with the blue bird, it makes green. If blending was correct, the F1 hybrids, these purple ones right here, they would always make purple because, like, watch this. If you took this plant, this represents many. If you took this and you self-pollinated it, what would happen if you self-pollinated it? Well, if you self-pollinated it, it would make a pod, and you'd have seeds. And these would be, if you're following this discussion, and I hope you are, these would be the F2, the second generation. When you plant these seeds, guess what happens there? 75% of the seeds grow up to have purple, and 25% of the children grow up to be white from these purple F1s. So the F2s are kind of a 3-to-1 ratio. Now, that is not random, and that is not blending, because it's one thing blending these together, but how can the white reappear in the second generation? So blending isn't the way it works. So Mendel, not knowing anything about DNA or chromosomes or any of the things that we now know today, in his journals, he was saying things like, like maybe there's some kind of factor, use this word, that's kind of a good generic term, there's some factor that determined purpleness. And there's a purple factor and a white factor. And he figured, if I may, let me sort of come off of this for a second. He figured that, let me call up this and do this. He figured, check this out, this is a great concept. He figured that if there is purple, like here, in this plant, and there is white over here in this one, he figured this is the parental cross. When you cross these two together, all of the F1 was purple. And so he started writing this down, and he was saying, this is interesting. It seems that one factor seems to always, keep the other one down. It seems to, one of my favorite words, it seems to dominate over the other factor. And so he wrote that down. He said that one factor in the F1 seems to dominate over the other. And then he said that, like in the F2, in the second generation, you get this 3-to-1 purple to white ratio. And so, his explanation of this was rather profound. He thought, and I'll come back to this because this is a very important point, but I'll just sort of a little foreshadow it here. He figured that, there was a purple factor and a white factor, and he figured that there must be two factors in each plant. We now know this to be homologous chromosomes. He figured there was a purple and a purple, which there is, and let me go like little p and little p for white. And I'm going to go lowercase because it's recessive. So this plant right here is the result of, this is a diploid, but the pollen from this is a haploid. So when you cross the purple pollen with the white pollen, with the white egg, what you get is a big P, little p. You put back to the diploid again. And this is purple. This is the purple ones. And what's interesting, this second generation, if I can do this, if you get a purple and a white crossed with a purple white, so these are two purple ones, and he figured that the factors segregated during pollen formation. And the reason that it was a 3-to-1 is that it would be purple, 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 and white. He began to see this. And so I will come back to this. You may know this somewhere in your past. This is known as a Punnett square. And these are representing the gametes up here, and this is showing fertilization. So this is where we're going. I just thought I'd point that out just because I know some of you have a background in this, and I just wanted to acknowledge sort of what you know and where we're going with it. And so, as I was saying before, the first generation is all purple. So what's fascinating is these F1 plants that were all purple, these F1s. So here are all the purple hybrids. Whoops, sorry. Purple hybrids. And so the purple hybrids right here can produce pollen, but the pollen that they produce, here's big P, little p, the pollen that they produce through meiosis is some of the pollen is purple and some of the pollen is white coming from this hybrid plant. Now if this was a pure purple plant, it would be purple and it would only have purple pollen. And so when you look at a white flower, the white flower produces only white. He figured this to be the case because every time you self-pollinate white is that it just produced white. Self-pollinated ones produce white over and over again, but when you cross the white with the purple in the first generation, let's just do that just to make sure. So the P generation, when you cross these two together, all the F1s were purple. And so the purple, we now know it as the purple allele because the trait is a petal color. So this is a purple allele and a white allele, then in the F2, what you get is a 3-to-1 ratio of purple to white in the F2. And so what Mendel did was he literally took the peas and he planted them and he recorded the results. And every time he did this, he saw the same pattern emerging. 3-to-1, 3-to-1, 3-to-1. And when I say 3-to-1, it doesn't necessarily mean like literally 4 plants. It's representative. It's going to be 75-25, or if you're doing 1,000, check this out. These are 705, 224. You're like, well, wait a minute. If it was 1,000, this should be 750, and this should be 250. Well, what you actually observe and what you expect may be slightly off. It's sort of like if you expect 5 heads and 5 tails when you flip a coin. The chances are it may not be 5 and 5 because you've only flipped it 10 times. But if you flip it many, 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 many more times, the true statistics come out. This is 3-to-1. And so what's interesting about this 3-to-1 ratio is that this is a pattern. This is a mathematical pattern that is predicted. Every time you do this, this happens. And so you're like, well, I don't know. Like, just because pea plants purple and white doesn't mean that it's anything of significance for me. Like, what does that have to do with more advanced organisms and humans and traits? Well, every time he did this, as I was saying before, the F1s were purple, and then when he crossed them, the white came back in the second generation. And if it was blended, it would never be able to come back. And every time he did this, no matter what trait he was considering, let's just look at some of these traits. I've been mentioning purple and white, and I've also been mentioning over here in terms of height, tall and short. But sometimes seeds are round and sometimes they're wrinkled. I don't know if you've ever seen these before, but there's round and wrinkled. It always seemed like the round one dominated over the wrinkled. Not that it means anything. Most of us are familiar with green peas, but some peas are yellow. And as it turns out, the yellow dominates over the green in the first generation. This is whether or not the flower grows off to the side or whether it comes off the top, or terminal or axial. And then pod color. Again, most of us are familiar with green pods, but there are yellow ones. And then inflated and constricted pods. So these are some of the traits that he considered. And what's fascinating is every time he did that, the F1s, one allele seemed to dominate and the other one seemed to be recessive. And so he wrote that down, dominant and recessive. And then finally, like, this is a picture of the round and the wrinkled. When he crossed this, this pattern of 75-25 kept coming up so many times. He began to consider, wow, it seems like every time, no matter what trait I'm doing, it seems like the first generation, these alleles always seem to dominate, and in the second generation, there's this 3-to-1 ratio. 3-to-1, 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 3-to-1. And so it's incredible. These are the actual numbers. And see how this one's very accurate, 3-to-1? And that's because when you're counting seeds, there's a lot more of them than if you're counting or just the whole plant itself. And so this was an amazing discovery. It may not seem like much, but we'll build upon it, that there's a pattern, and the pattern is emerging, and where there's a pattern, there must be some really cool reason behind it. And so I hope this was enjoyable and that you found it interesting and it was informative. And so thank you very much for watching.